So the second type of lymphocytes are the T lymphocytes. So before even looking further into the chapter, let's first see that why do we need lymphocytes and how are the difference. So first of all, let me tell you that uh, let me tell you this that so the B lymphocytes, as we saw, like after well they were forming, like the outside of the bone marrow, they divided when they were like stimulated by pathogens. Like if you were infected and there were pathogens outside, so they stimulated and they further divided. But what happens in T lymphocytes is that not the pathogens antigen directly influences the production of T lymphocytes, but they act when antigen presenting cell. So this antigen presenting cell is actually the phagocyte. Like we saw WBC are of two types. Uh, so this WBC was of two types. One was the phagocytes, the cell ingesting, the physical ones, and one was the lymphocyte, which is T lymphocyte, which is part of the lymphocyte system. So when phagocytes, when they digest, like when they're ingesting it, and they've ingested it, and they've killed the pathogen, so that antigen come out of the cell and they might stick out like this, and that thing we can call as antigen presenting cell. So when this phagocyte is acting as antigen presenting cell, at first they have already done it, like they have already killed the pathogen. So after it has done it, so we know that so like for the process to get more faster and to help the phagocytes, this triggers the T lymphocytes to um, this triggers the T lymphocytes to be produced. So T cells cannot bind directly to the pathogen. Instead, they bind to the antigen representing cell, which is this. So it's the pathogen. I mean, it's the phagocyte, but uh, it's these that stick out of that pathogen. I mean, out of this WBC, the phagocyte, and this whole acts as antigen representing cell. So this cell, I mean, triggers the production of T lymphocytes. This is the first difference, and the second one is like uh, they're similar in this sense that they also have like so they have two parts. I mean, there are two types. One is the helper T cell, and the other one is the killer T cell. So so the another difference you will notice, like let me proceed first. So let me tell you the difference first. So we don't see the production of antibodies here. We see different chemicals being produced, which are like uh, more destructive. For instance, the killer cells produce cytotoxic. They release cytotoxin. So that's a chemical itself. So which is more which is more devastating and more effective. So there are two types. First of all, there is the helper T cell, and then there is the killer T cell. So these helper T cells, so once the T cells are produced, uh, like they like to work in combination, like because the pathogens they divide so faster that they always need each other's help. So the phagocytes, I mean the what am I mean like the yeah, the phagocytes. So the phagocytes triggers the production of T lymphocytes, and then the T lymphocytes produce helper T cells, which can divide into two. One is the memory and the helper T cell. So the function of the helper T cell is that it helps to uh, it releases cytokines which promotes the plasma cell formation. So the helper cell produces a chemical that helps to produce more. To they go to the parent B lymphocytes and they like they help the production of more plasmas. They trigger the more production of the P lymphocyte cells. And the cytokines also promote like the T killer cell, which is this we'll talk which we'll soon talk about. And cytokines also promote vigorous phagocytosis. So also the how the phagocytes do their process, ingests. So the helper cells actually what they do is like they help the other type of WBCs to contribute more towards the killing of the pathogen. So this is the helper cell, and then we come across the killer cell. So the way that it is different, because the killer cell is also like producing such chemicals that kill mm, the pathogen, but these chemicals are not antibodies which B lymphocytes created. So so the killer cells are of two types also. I mean, they can either be killer T cells or the killer T cells itself. So here also we get memory, and we get these memory cells because we already say that this cannot further divide, but the memory cells can. So they can create more memory cells and more helper T cells. So these are the active forms, and these are the memories which are there because they can divide, but this can divide. So that's the importance of the having memory cells of their counterparts. So these T killer cells. So this function of the T killer cells also makes T lymphocytes different than B cell lymphocytes, is that they don't produce antibodies. Rather, they release a cytotoxin, which is a toxic substance which destroys pathogen. Uh, Likewise, antibodies, they have a range of activities they can do, which leads to the destroying of the pathogen. But here, we produce an alternate, which is the cytotoxin. So this killer cell produces these chemicals called cytotoxin, and they destroy this pathogen. So they are dead or destroyed, and this is the difference. And so moving on, we have that I have a note about why we need memory cells. So as I told earlier, the counterparts, they divide into two, and they always have this memory cell and this killer cell also. When they divide actually, what I'm saying, like uh, the memory cell can only divide. So that's the importance of having uh, memory cells. Like when a memory cell divide, uh, for instance, this killer cell, it after it has done its job, it will live for temporarily, and soon it will be dead, and it can't further divide. So what about the time of reinfection? We talked about that in the second time it will be faster. So if it's dead, so how's that gonna help? So uh, but the memory cells of the T killer cell will be present. So that can produce further memory cells as well as the T killer cell itself. So, and again from that memory cell you can produce more memory cells and also the killer cell itself. So we see that this is acting as a database for producing the killer cells, and as the killer cell is not living for that long and memory cells can live for that long. So that's how it's working together. So moving on, so let's have a summary. Let's look at a summary. So WBCs are of two types. One is the lymphocytes and the phagocytes. So phagocytes are the ones which are cell ingesting. So they are more physical. So one is the macrophage and the neutrophils. So neutrophils are smaller and the macrophages are a little larger. And the nucleus are connected, and nucleophiles are found everywhere, but macrophages are only found around organs. They usually work when neutrophils are failing. So moving on, lymphocytes, which are more chemical based, and they work by releasing chemicals such as antibodies or cytokines or cytotoxins. So there are of two types. One is the B lymphocytes and the other one is T lymphocytes. So the B lymphocytes are more activated when the helper cells are released, which was activated by the phagocytes. So uh, for understanding the activation process, we have to look at the whole range. But now let's just look at the divisions of each uh, cells or the types. So the lymphocytes are of two types: the plasma cell and the memory. So we see that every this is the active form which acts, but with this active form and this active form and this active form, we always see there is a memory cell being created. uh, let's mark it with green. So here's the memory cell. I mean, T helper cells also has its counterpart memory cell, and this killer cell also has a memory cell. So why is this? We already discussed that the plasma cell, this is the working cell, but they cannot further divide. So memory cells can further divide into memory, which can again further divide, but while division they can also produce a lot of plasma cells, and same for all. So moving on, then so the plasma cells work by producing antibodies. Now coming to the T lymphocytes. So T lymphocytes are activated when the phagocytes, for instance the macrophages, uh, had ingested pathogen with its antigen in it. So after ingesting it and destroying it, it may have its the pathogen's antigen on its surface. So it can act as uh, by like an antigen representing cell. So T lymphocytes are activated by that way, and they are of two types: T helper cells. So as I said before, that the helpers are like I mean, they like ask for help, and they are like triggering other cells uh, be, to be like helping them more. So they trigger the production of the other cells. So the helper T cells that we saw earlier, they uh, promote more B lymphocytes production. So by releasing a chemical called cytokines, as I wrote here, on activation by cytokines, and they also trigger the more production of phagocytes itself. But ironically, it was triggered itself by phagocytes, which had an antigen itself in it. And key T cells are like the plasma cells, but the difference is that the T killer cells do not produce antibodies, and something similar to antibodies, uh, which are specific substance which we call cytotoxins. So these are chemicals which actually destroys the pathogen. So this will be we have finished the lymphocytes and the summary, and this